All right, TSA. Why do we do the transcriptome shotgun assembly TSA database limited to Lambdotia punctata? Like, why do we do this at all? All right. So this is a blast N of our cDNA. That's the cDNA from PC1. The subject is the whole genome of Lambdotia. That's why it's the limiter was Lambdotia. The limiter was Lambdotia. The whole genome. So think about that. That's a cDNA against the entire genome. The cDNA, which is a messenger RNA, against the whole genome. Why is that interesting? Well, if you look at humans or plants or any eukaryote, pretty much, I'm generalizing, but it pretty much holds true. Not so much bacteria, but pretty much any eukaryote. Your cDNAs are minor compared to your whole genome in terms of scope and size. You see, our cDNAs, which are messenger RNAs, we don't have cDNAs, we have messenger RNAs. We make them into cDNAs when we do biotechnology on them. But your cDNAs, which are messenger RNAs, that's your exons. That's your expressed DNA. That's the genes that are being expressed into proteins, usually. Sure, 20% of your genes are introns. They actually get spliced out by things called SNRPs and spliceosomes. We'll talk about that later. But the cDNAs are expressed. The cDNAs are expressed. That's your hair. That's your skin. That's your eyes. Right, the proteins. That's your pancreas protein, your liver protein, your gallbladder protein, your blood proteins, your bone marrow proteins, your immune proteins. All the stuff that we can see is your exons, your messenger RNAs. They're in messenger RNAs. But look at the everything else. Look at the green. Look at the medium green. Look at the, I don't know what color that is. Forest green? I'm not really sure. Look at the blue. Right, all of this, that's roughly 75%. Whoops, that should be a 7. That's roughly 75%. So 3/4 of our genome is just code. It's just code. It's called intergenic code. It's between the genes code. Sure, there's evidence that some of it was old genes that are defunct, no longer used. But most of it is repetitive DNA. Repeats, repeats, repeats. Now, where does that repeat? Where do those repetitive DNA come from? Transposons. And transposons come of two types. DNA transposons, which do a cut and paste. But the real significant ones, I think, are the retrotransposons, which are RNA-based. Well, they're DNA, but they're made into RNA, which then reverse transcribe back into DNA, and they copy and paste into your genome. These transposons result in these massive repeats of code in our genome. Again, this is not genes. This is just code, code, code. Now, why do we have these? Where do we get them from? We don't really know. But the strongest theory is that we get these from viruses. The strongest theory, that with the most support, is that they come from viruses, because this is what viruses do. This is what viruses do. Viruses, a lot of them, like herpes, like HIV, like pox virus, like papillomavirus, like cytomegalovirus. All these viruses, you may have heard of those. I know you've heard of herpes. I know you heard of HIV. All of these copy themselves and insert themselves in our genome, and they hide in our cells during your life, and we die with them. And they're ours, and that's it. And they just fatten up our genome, if you will, during our lives. They make our genomes larger. That's what comprises most of our genome, 3/4 of our genome, roughly. These are called transposable elements. Transpose means to copy. Now, yes, it's like really 40-ish% are definitively transposable. But if you look down here, these are unrelated, but we just don't know where they came from. And these, we really don't know where they came from. They could be transposons. It's just we don't have any evidence for that they are. Wow. So that's a lot of our genome is just copies, copies, copies. Now, I mentioned those viruses before. HIV, herpes, cytomegalovirus, all these pox viruses, all these viruses, we know about these because we've found them, we've crystallized them, we've seen them, we've sequenced them. They cause actual disease. But it's estimated that for every one virus that causes a disease, there's many out there that don't cause human disease that we never even know about. I could have picked one up this morning, right? I could have picked one up this morning. I don't know. And it never causes me any disease. And it just forms a transposable element and replicates in my genome. Maybe. So we think we get these during our lifetime. Lastly, all of this, the 75%, that's a lot of code. And companies like 23andMe and Ancestry.com, those genome sequencing companies, they do a lot of your genome, and they use this 75% because humans either have them or they don't, or they have them in repeats. So I and my two brothers, my relationship, my siblings, right? My related siblings probably have a lot of the same repeats because our genetic line have picked up a lot of these repeats. Do they make us look a certain way? Probably not. Do they make us behave a certain way? Probably not. I look like my brothers somewhat because we have the same, the purple. You look like me. You do. All humans do because we have the same genes. All humans have the same basic genes because we're all living humans. We have very different transposable elements. And we think it's because the transposable elements we pick up during our lives and they replicate and replicate and replicate and move around our genome a lot. And as a result, they're very mutable. They're very mutable. Whereas the purple, the actual genes, coding for proteins, those are mutable, no doubt. But if you get great mutations in them, you're not you. You don't survive. Massive genetic mutations usually result in death. Chromosomal deletions, really bad gene mutations like, I don't mean to be insensitive, like Tay-Sachs, for example. These are single gene problems or chromosomal problems that can result in a non-living human. So the point is, if you're a living human, you have all these genes, these purple genes, if you will, the actual messenger RNAs from the genes. And loosely said, it doesn't, your 75% doesn't really matter probably. So that's what the TSA is searching, the whole genome, this whole pie chart. The TSA is searching the whole pie chart. It's taking your cDNA of PC1 and relating it to the entire genome. All right, I'm going to stop recording there. Bye-bye.